Alright guys, so we are starting our last official topic of the year. I know that's probably a good feeling. We are in the final countdown. So we just have a few more lessons to go on Kingdom Animalia. And so we're going to kind of be introducing animals today. So just to kind of give you a heads up, I know there's a lot going on in the spring. But we will be doing semester exams here in the next few weeks. Everyone is going to take their semester exam when the seniors take it. That way I can get all of my grades turned in and everything on time. And really all we have left to do is we have one more unit test, and then you guys are going to be getting ready for that semester exam. So we'll try to get those semester exam reviews out here in the next couple of weeks, and that way you guys will have plenty of time to prepare for that. Hopefully we can get everything wrapped up and you'll have some time in class to work on that as well. So the last and final thing to cover is Kingdom Animalia. And hopefully of all the kingdoms, you are more familiar with animals than any other because you are one. And that's something that's very important to understand. Over the years, I've had students that did not realize that they were an animal, and that's a little bit problematic. And so we need to first take a look at what makes an animal an animal. Okay, so number one, they are multicellular. We do not have unicellular animals. I can't think of any exceptions to that rule. And pretty much if they were animal-like but unicellular, they'd probably be going into one of those protist groups. Another characteristic is that all animals are heterotrophs. Animals are not able to make their own food. Of course, we're still in domain eukarya, so they all have eukaryotes. And then another important one, something that we've seen over and over and over again, is cell wall. And so for many of the other kingdoms, it was about the composition of their cell wall. But for animals, it's simply that they don't have one at all. And so since they do not have cell walls, they're going to have more stuff in their extracellular matrix, which this term literally means outside of the cell. So the matrix is just the carbs and the protein fibers that you will find outside of the cells. And that's going to help provide more structure. Many of your animal cells have unique cell junctions. We actually talked about these a little bit back when we covered cell transport, I believe it is. Or maybe it was just cells in general. I think it was just cells in general. But we talked about desmosomes, which are kind of like spot welds or buttons or snaps. They're kind of like little areas where the membrane is fused together with some proteins. There's tight junctions, which kind of form seals, like in places where we don't want leakage. And then there's gap junctions, which are essentially little channels for communication. But typically animals will have these cell junctions that are holding their cells together. Now notice here, this one says most. Okay, so this isn't necessarily an all, but most have tissues. And so when we talk about the organization of life, we know that cells make up tissues, tissues make up organs, organs make up organ systems. And so for the most part, most of your animals are going to have these four types of tissues. And we're going to be covering that in another set of notes, and we also have a lab over our tissues coming up probably later in the week. Most of your animals are sexually reproducing. However, there are some that do have some asexual methods. And then one of the things that's unique are Hox genes. We actually saw this back with gene regulation. We also saw Hox genes when we were covering the Evo-Devo unit. And these are essentially just like body plan. These are body plan genes. These are genes that turn on and regulate other genes. And so in terms of what makes an animal, here we have all of our major characteristics. Now in terms of animal development, we've actually covered this a little bit in the past, but to kind of in order to understand some of the ways we classify animals, we do need to kind of understand some of these early developmental stages. And so the animals are going to begin as a zygote. Of course, we're talking about sexual reproduction here. So we have that diploid zygote. That zygote is going to undergo rapid cell division. Now they're actually called cleavages, and they divide, but they don't separate. Okay, so notice we don't have cytokinesis. So they're dividing, but they're staying together. And they get to the point to where they form this hollow ball of cells called a blastula. So kind of think about a basketball, and then think about a basketball that needs more air. And what happens is this hollow ball of cells, this blastula, begins to kind of cave in on itself, okay? And that process is called gastrulation. And this is going to essentially be the beginnings of your digestive tract, okay? And then through the process of gastrulation, if you notice the colors here, what we get are these layers of cells. And then we have this opening known as the blastopore. And so in terms of classifying animals into different groups, one way we do it is based on whether or not they are diploblastic or triploblastic. Now, most animals are going to be triploblastic, okay? And what that means is they have all three of what we call these embryonic germ layers. So if we go back, you see this hollow ball as it kind of pinches in, we have these layers, and these layers become what we call our embryonic germ layers. And so we typically have three. There are some animals, your simpler animals, are going to be diploblastic. They only have two. They don't have the mesoderm. But we have the ecto, the endo, and the mesoderm. And so these words are pretty easy to understand because we know that ecto means out or outside, endo means in or inside, and meso typically refers to middle. So the outer layer is actually going to become the integumentary system, the skin, and it's also going to become the nervous tissue, which this one right here is pretty easy to remember because the skin is on the outside of the body. The endoderm is going to form from that innermost tube, and so this essentially is going to become the digestive tract and kind of the smooth muscles of some of your basic kind of inner cavities. So this is going to become that hollow tube that kind of runs through the center of your body. So it's going to be inside. And then the mesoderm is just going to be the stuff in the middle. So all of our connective tissues come from the mesoderm. So things like bones, fat, blood, all of those are going to come from the mesoderm. And then we also have all of our muscles are going to come from the mesoderm. So the ecto on the outside becomes skin and nervous, the endo on the inside becomes the digestive tract, and then the mesoderm is going to be a lot of the middle layer stuff. And so we as animals are triploblastic as well as most other animals. Another way we can group animals has to do with that blastopore. So the blastopore was the opening. So when that gastrulation occurred, we kind of have this opening to what's going to become the digestive tract. And so depending on the type of animal, that blastopore is either going to become the mouth, or it's going to become the anus, okay? And so what we're left with are some animals are protostomes. Normally your simpler, earlier animals are protostomes. And then we have two that are deuterostomes, and that's going to be your echinoderms, which are things like starfish, and that's going to be your vertebrates, which are going to be your fish, amphibians, reptiles, birds, and mammals. So your most complex animals are going to be deuterostomes as well as your starfish. Now, I know it kind of seems, well, duh, but, deutero, doo-doo, anus, I mean, it's kind of easy to connect those two. So in a protostome, the mouth forms from the blastopore, and that's pretty much going to be almost all of your invertebrates. That's going to be your insects, that's going to be your clams, it's going to be your jellyfish, all of those, your worms, and then your deuterostomes, the anus forms from the blastopore, and that's mostly going to be all your vertebrates. And so there's some other things that differ between the protostomes and the deuterostomes. So when you look at like the cleavage, remember this is the cell division, so you can see how the pattern is slightly different. Okay, this is called spiral, and that it's kind of spiraling in on itself, and this one's radial, and that it's more circular of a body plan. Determinate means that very early on those cells know what they are going to become, and indeterminate means later in development these cells kind of are told what to become. So when we were talking about like cell specialization, these take a little longer to know what type of cells they're going to be. There's also a little bit of a difference in the coelom, which is a cavity between the protostomes and the deuterostomes, but really the main difference is what's forming from that blastopore. All right, animals can also have a different body plan. Now, pretty much there's only one group of animals, that's your sponges. Those are your simplest animals. They demonstrate asymmetry, which remember if you put an A or an and in front of something, it means not. So these are the group that do not have symmetry. You can't really fold them in half and get two equal halves. Some animals display radial symmetry, which when you think about radial, I want you to think about a circle, a radius. So radial symmetry are those that have a circular body plan. That includes cnidarians, which are going to be like your jellyfish, your sea anemones. Some of your starfish actually have what's called a 5-point radial symmetry. It's not a perfect circular body plan, but it also does have more of a radial type. But most animals are going to display what we call bilateral symmetry, and this word literally means two sides. Bilateral means two-sided. And so these are the ones that have a left and right side. These are the ones that are going to typically have a top and a bottom. They normally have a head, okay? They're going to have cephalization. They're going to have a head on one end and possibly a tail or another end on the other side. They're going to have left and right. They're going to have paired structures. And this is going to be most of your animals. So there we have the radial. Pretty much you have multiple lines of symmetry when they have that circular body plans, whereas when you have bilateral symmetry, there's just one line of symmetry that's going to divide them into left and right. We can also take a look at animals in terms of whether or not they have a body cavity, and the word for that is a coelom, okay? And the purpose of a coelom is it normally is fluid filled, and it's going to cushion. It's going to sometimes act as a hydrostatic skeleton. Remember that since animals don't have cell walls, they need some type of support. It's also going to allow those organs to kind of be suspended and cushioned and allow them to move more independently. And so we have three groups that animals could fall into. We have coelomates, we have pseudocoelomates, and we have acoelomates. So once again, we see that A, which means not. So these are the ones that have no body cavity. This is essentially going to be your flatworms, your platyhelminthes. They're pretty much tissue on tissue on tissue. They don't have a space or that gap in between their tissue layers. Now, pseudo means false. And so they have a cavity, but it's not considered a true body cavity. It has the cavity itself is not completely formed from the mesoderm tissue, and I'm going to show you a picture here in a second. It's just going to be a space between the endoderm and the mesoderm. Whereas a true coelomate has a fully lined cavity that is fully surrounded by that mesoderm tissue. And so let's take a look at our picture. So this right here, this is a true coelomate here. Okay, they have the prefix U in front of there, which means true. But a coelomate, see this red tissue? Okay, notice how it's going all the way around the space. So here's my cavity, here's my coelom, and we can see that middle layer tissue is completely surrounding it. Whereas down here, there's my cavity, this kind of fleshy looking mesoderm tissue, it actually doesn't line the whole cavity. So this is a false cavity. And as far as I know, this is mostly your roundworms. Most of your animals are going to fit in this category up here. And then here you have your flatworms, your acoelomates. So this would be the digestive tract. That's not a cavity, but you can see that there's no space. It's pretty much tissue on tissue on tissue. All right, some other trends that we're going to see with animals is cephalization. Earlier I mentioned that with bilateral symmetry, the animals had a head. And so cephalization is the head formation. It's essentially where the sense organs get concentrated at one end, the anterior or the front end of the body. It's going to allow for more complex senses, more complex response, more complex thinking. And so as you go through your animal groups, like when you do your lab, you're going to see that cephalization typically increases. As the animals become more complex, they are more cephalized. And so the more complex the animal, the more cephalized they are, and the greater their ability to respond and sense and think, okay? By the way, the prefix cephal, in general, cephal means head. All right, we can also group animals based on how they regulate their body temperature. Okay, so we have thermoregulation, and we can put these into groups as either endotherms or ectotherms. Now, growing up, you probably heard terms like warm-blooded and cold-blooded. Those aren't necessarily the best terms to use because cold-blooded can be a bit deceiving. Cold-blooded animals don't necessarily have cold blood. Okay, now really the difference is how are they regulating their temperature. If it's an endotherm, and these are the ones that we would typically call warm-blooded, if it's an endotherm, they have an internal temperature maintenance. They're going to have lots of mechanisms that help them maintain a relatively steady internal body temp. And this is going to include us, all mammals, and birds. Okay. Now, in order to maintain that high body temperature, they're going to have a high metabolism, which is constantly generating heat. They're going to have insulation, like fat. They're also going to have those negative feedback mechanisms. So, in order to maintain that internal body temp, we have to have some mechanisms in check to help us not get too hot or not get too cold. Now, the difference with ectotherms, once again, cold-blooded is really not very accurate because the key to ectotherms is their body is affected by the environment. So if the environment is cooler, then their body temp is cooler. If the environment is warmer, then their body temp is warmer. Now, they still have to stay within a range because extremes could be deadly. And so this is why when you look at like ectothermic animals like invertebrates, fish, amphibians, reptiles, you don't really see them out in the wintertime. Okay, they're normally kind of buried down under the ground in order to stay warm. Or when it's really hot, you will see them in the shade. You know, you won't really find them up close to the bright light or to the direct light. Or if it's just a nice spring day, you may see them bathing in the sun, kind of just chilling. And so ectotherms, they have a much greater range of temperature. So here we have our endotherm. We can see it's a very narrow range, whereas the ectotherms kind of can vary over time, depending on the environmental temp. All right, so what kind of organisms will you find in animals? We've kind of mentioned them a little bit, and this is what your lab is going to cover that you're going to work on. We're going to look at, I think, 9 different phyla. We won't be looking at Rotifera. Those are rotifers. We won't be looking at Lophophorids. I don't know a whole lot about those. We won't be looking at Nemertea, but we will be taking a look at all of these other phyla. So we have kingdom Animalia, phylum Porifera, phylum Cnidaria, phylum Platyhelminthes. You will need to know these for lab quiz purposes, but hopefully you're going to get a pretty good exposure to some of the different groups of animals. Now, of all of our animals, about 95% are invertebrates, which means they do not have a backbone. And so here you're going to have your porifera, which are your sponges, cnidarians, which have stinging tentacles like jellyfish, platyhelminthes or flatworms, nematoda or roundworms, annelida are segmented worms, arthropoda are things like insects, mollusca are things like clams and squid, and then echinoderms are your starfish. They're kind of your spiny skinned invertebrates. So this is what you're going to be taking a look at in your lab, and you'll get more details on those there. And so here's kind of the cladogram, the evolutionary breakdown of what they have and what they don't have. So like here you have some protostome groups. There you have your pseudocoelomates, your acoelomates. You can see your symmetry, radial and bilateral. So there's a lot of important information here on this chart just to kind of give you that general breakdown and to kind of tie in some of those things that we just talked about. And then the rest of your animals are chordates. Okay, so notice we didn't just say vertebrates, they are chordates. And chordates are animals that at some point have these 4 characteristics. Okay, they have a dorsal hollow nerve cord. This is essentially going to become the spinal cord in the more developed animal. They have a notochord, which is a supporting rod. That is essentially going to become the spine or the vertebra themselves in most animals in your chordates. The pharyngeal or pharyngeal clefts or slits, these kind of look like gills in an embryo. For us, they become part of our inner ear, but for some animals, they do become actually gills. And then we all had a tail at some point, okay, as an embryonic stage. Some of them keep the tail, some of them don't. But in order to be considered a chordate, so this right here is what separates the invertebrates from the chordates, as the chordates have these 4 characteristics. So here we have that dorsal hollow nerve cord, we have the supporting notochord, then we also have the pharyngeal slits or the clefts, and then we have the tail. Now when it comes to chordates, all of your vertebrates are going to fit in this category, okay? So when we're looking at phylum, we have kingdom Animalia, phylum Chordata, and then subphylum Vertebrata. So not all of your chordates are vertebrates, but most of them are, okay? These are going to be your fish, your amphibians, your reptiles, birds, and mammals. So we are in phylum Chordata, subphylum Vertebrata. Now, there are a couple of what we call non-vertebrate chordates. So they're not invertebrates because they have those 4 characteristics, but yet they don't have the true backbone to be a vertebrate. Okay, so they have the 4 characteristics, but they don't have the true vertebra. And that's going to be your tunicates and lancelets. They really kind of look, lancelets look like a little tiny fish. You should be able to see one of those in the lab that we're doing. And then tunicates, they kind of look like just like an aquatic, almost like a sponge type, but they are in the chordate category. And so here you can see the different groups. There's your tunicates, there's your lancelets. Okay, these are your non-vertebrate chordates. And then everything else is going to have your vertebra. You have jawless fish, cartilaginous fish, bony fish, and then you have amphibians, reptiles, birds, and mammals. And a lot of these guys are in they're in their own class. And that is going to wrap up our first lesson on Kingdom Animalia. Once again, you're going to be doing a lab coming up. I also want to just take just a quick second and talk about, if I can drag this onto the screen here. I want to talk about your study guide for a moment. So in your study guide, you have some more practice for animals, but I wanted to point this out here, okay? In fact, you're going to have a day to work on this. You could possibly start it today if you want to, but this is, notice it says a note sheet. So this is part of your animal study guide. It actually comes from chapter 40 in the Campbell book, okay? So we had a big book that we used to use for AP Bio, but honestly, you could find this information from anywhere. You could Google it, but this is some information that is not going to be in any other set of notes. Some of it will be, like I'm going to give you a set of notes on tissues for the lab, but like some of your homeostasis, we've looked at that before, but that's just a review. Bioenergetics, you're not going to see these words anyplace else in some of these areas. So I just want to go ahead and give you a heads up that you do want to do this set of notes. At least these open-ended questions here that may not be somewhere else, okay? So I would encourage you to maybe today get started on that, or when you have a day to work on them, please actually get these done because they are a set of notes that you need to know. In terms of your essay for animals, it's a jellyfish life cycle. It will be included in your study guide, and you will have a day where you do the plant and animal essays, and you're going to have one that you have to write about the plant guy, and then you're going to have 2 diagrams that you have to label, the moss life cycle and the jellyfish. So that's all I have for you guys today. I hope you have a good one, and I hope to see you soon.